while they're doing that, just like to echo Catherine's remarks about the growth of this big brain community, which has now become formalized as the highball community. I think we all saw yesterday the excitement that this engenders across our partners internationally. This has been quite a journey for the last close on 12 years that we've been working together on the big brain, and it's a delight to see the expansion that's taken place in that time. It falls to me to welcome our Sievers lecture today. We're delighted to have as the Sievers lecture in computational neuroscience, Professor Mu-Ming Poo. He will be speaking on the transcriptomic, connectome, and neuromodulation of the primate brain. Mu-Ming Poo is a giant in our field. He's a member of the Institute of Neuroscience of the Chinese Academy of Sciences. He's a member of the Chinese Academy of Sciences, Hong Kong Academy of Science, a member of the US National Academy of Sciences, and he's an awardee of the Gruber Neuroscience Prize in 2016. This talk is made possible by a generous support from the Sievers Computational Neuroscience Initiative that provides support to a lot of our activities in the highball field. It brings together cross-disciplinary experience in neuroscience research and neuroinformatics, computational modeling of brain states in healthy aging and disease, training and mentoring for tomorrow's researchers, and computer infrastructures and brain-based data sets to support this research. So with no further ado, I'd like to pass the Zoom link over to Professor Mu-Ming Poo.
Thank you, Alan. I would like to say a few words before my lecture. I thank very much Alan's and Catherine's invitation to this big brain workshop, and I look forward. I have been looking forward to this trip to Croatia, but unfortunately for this returning to China quarantine prevented me to do this. So I'm still very glad to be able to communicate through this video conference. So my talk today will begin with an introduction of the China Brain Project. The Chinese brain community are looking very much forward to the participation in the big brain activities. And so I'm sort of a representative for many of my colleagues here in Shanghai and in other parts of the Chinese in China. So my talk today will begin with a short introduction. So I would begin with the introduction of the China Brain Project, which was initiated last year. It's a big science project for next 10 years. The main project the main body of the project focus on the basic neuroscience, looking at neural circuit basis of cognitive function, and as well as the cognitive development. We are setting up nationwide brain research technology platforms, in particular for mesoscopic brain mapping. And this core of basic study will serve a purpose for applied neuroscience into brain-machine intelligence technology that includes brain-machine interface, neuromodulation technology, brain-inspired machine learning, and brain-inspired computing devices. And for the brain disease, we also hope to help the basic science to help the pathogenesis mechanisms for major brain disorders, and developing new diagnostic and intervention methods, especially for early diagnosis and early intervention. And we are setting up large community and patient cohort in order to facilitate this diagnostic intervention studies in the laboratory. The mesoscopic connectome brain and transcriptome analysis is the main focus of our brain mapping. Clearly, this is not only for the basic research, it's also very important for our brain disease and brain intelligence technology aspects. This is driven by the recent development in many of the mesoscopic technology, which will be my focus to discuss today. We would like to know for the big brain, for macaque and the human brain, the whole brain atlas of these cell types. This is obviously of interest for many laboratories throughout the world, in Europe, in US, and Japan, all the places. The recent development focus on the mapping of gene expression in all the brain regions, the so-called transcriptome, especially spatially resolved transcriptome analysis. The hope is to be able to identify all the cell types through the sequencing and through the identification of the specific cell markers and their regulatory elements. Once we the so-called enhancer and promoters for the specific genes, using this we can begin to address the structural connectomes at the mesoscopic level. We would use these specific labels to map the long-range projections in the brain, the input and output connections for each type of neuronal type identified in all the brain regions. Now, this is clearly being carried out in mice with quite a success. We are now hoping to extend this structural analysis to monkeys and maybe eventually to humans. Now, there is also a problem of local circuits, which are very difficult to study at the light microscopy level for the various brain regions. So microscopic analysis at EM level would be very useful to facilitate the local circuit analysis, especially EM reconstruction, EM technology that involves the ability of labeling specific cell types. The development of the structural connectome will lead to further functional analysis at the circuit level. New technology for monitoring and manipulation of brain activity can be developed. Once we have a cell type identification and specific cell marker or the regulatory elements, we can express the probes for activities and for manipulating the circuit activity. This is the goal. The recent developments in the in the cell classification by gene analysis is based on the long history of cell type analysis. We know the agenda began with Cahall's mapping of the cell type by morphology, and the last couple of decades, the mapping of different neurophysiological defined cell types in the many laboratories are working on in monkey, as well as human brain for single cell characterization. These three classification scheme is now further extended to a classification based on the projectome. Now, in the mice, at least, one can now classify the cell type based on a more defined projection, their target, as well as their axon branching pattern, as well as their cell morphology, including the dendritic morphology, which is mostly focused by Cahall many years ago. So this the cell defined morphological identification of cell types now can give you another criterion for a classification of cells. So all these four cell type classification needs to be integrated. At present, the effort for integrating the morphology and physiology has been pursued, as well as the transcriptome has been pursued in several laboratories. But the matching of the cell types defined by these three different methods with the projectome map has not really begun. Because the information on the connectome is at present is still incomplete and sparse. So I would like to see to talk about this issue of cell typing as a main focus for my talk today. How do we define cell types? What is the criterion we need to follow in order to determine the cell? Because this is basic for understanding of the specific circuits in the brain. We need to know the cell type. Based on the rapid advance of transcriptome analysis, one can now have a complete atlas of the cynomolgus monkeys. For example, recent paper appearing, a large collaboration initiated by BGI in Shenzhen, China, with many international collaborators. They published the complete atlas for 43 tissues of the macaque, cynomolgus monkey, and using cluster analysis. And this is the dissociated cell doing single cell analysis. They can define at least more than 100 types cell clusters, so we can define the cell types in various tissues. Now, this clearly is an underestimate of the number of cell types, because we know even in the brain, there are hundreds of different cell types can be distinguished by the transcriptome analysis. Now we can now begin to do this with the macaque brain. So recently we have accomplished seen a large number of cells, hundreds of thousands, more than 1 million cells from the cynomolgus brain to do the clustering analysis and define major cell types, glutaminergic, GABAergic, non-neuronal cell types, and define these cell types based on the markers we have known previously in study with human brain as well as a mouse brain, as well as based on the location in the brain, their brain region locations. And based on all these properties, features, we can now classify a large number of the cell. We call these cell clusters rather than cell type because we are still clearly not sure about all these different excitatory neurons. Are they really defined cell type, or they are simply reflecting different state of the cell in that particular time when we do the analysis? And are these stable cell types? That remains to be studied. So at present, I think the best to call all these clustering analysis cell clusters, cell clustering or classes, rather than specific subtypes of the excitatory neuron or inhibitory neurons. All these have different distribution. Based on these criteria, they have different distribution in different brain regions. The number of cells of number of cells in different brain regions, major brain regions, are quite different. So clearly, this is something that needs to be analyzed further. Now, this is the simple standard transcriptome analysis where the cells are dissociated from, at best, from specific region when we dissect the tissue. So we know the origin of these cells coming from different brain regions as defined anatomically. But we really want to know is in situ gene exon analysis that's now coming into the scene in the last 2 or 3 years. Now, spatial transcriptomes involve mapping the gene expression on brain slices. Now, we have completed recently a few monkeys with complete spatial transcriptome analysis. Now, this is an example of a section of coronal section of 200 sections from the brain, and you put these thin slice brain section, 10-micron sections, on top of a chip. Right, the chips could be large chips, 3 by 5 centimeter chips. The chips is loaded with nanoballs, with 150 nanometer nanoballs, 500 nanometer spacing. Nanoballs are all linked with DNA barcode for their position, and as well as poly T, so that they can capture poly A mRNA, poly A tails of mRNA on top of these chips from the slice, the thin slice that lay flat on the chips. From there, we can get the so-called stereo-seq information of all the gene patterns. On the at a single cell resolution, you can look at the individual cells, and you can look at an expression of mRNA's clusters, and you can scan a single-strand RNA, DNA for their location of the cluster of transcripts, and then you can do single-cell segmentation, a method which is automatic methods for segmentation of single cells. And now help us to obtain up to 1,000 genes per cells from the tissue. Now, how are these cell type determined? We have to use the transcriptome, traditional transcriptome analysis. So what we do is we have at each section we have a set of sections cut from each brain regions, including thin sections for spatial transcriptome analysis and thicker sections for snRNA, single nuclear RNA sequencing analysis. We have a large number of cells, and these sections for RNA-6 are dissected from this slide, so we can know their regional locations. From there, we can do a single-cell clustering analysis to define the cell type, as I described earlier, based on the traditional transcriptome analysis, and then match these cell types with the all these single cells, what type that they could be. And this will be then further confirmed with another slice, adjacent slice for immunostaining, for confirming the gene we identify are really authentic. So this would allow us to obtain this single cell resolution spatial transcriptome. Now, for segmentation approach, where we can do we can collect the RNA, right? So some of them are clustered together, so it's hard to separate this RNA. We can do total RNA, the single cell are harder to identify. So we use a technique of a single-strand DNA scanning, which will show better the nucleus of each cell. Now here, we still don't see the boundary of the cell, so we added a membrane staining, the connectome staining with standard cell membrane. All three information together, the RNA clusters, ssRNA staining, plus connectome staining, then use the automatic segmentation, the machine learning method to segment each cells, and this is developed by Tencent AI lab. So that would achieve a pretty good, highly reliable single cell identification. So from there, we can have a single cell resolution for the entire cortex. You can see different cell type distribution, which is region specific, as well as layer specific. This gene expression pattern can be used when starting to use this. Look at the overall gene expression bundle. Pattern changes from region to region. There are a sharp boundary that can be identified between regions of gene expression. That will help us to get additional information from anatomical or histological staining defined boundaries. And there are many of these boundaries are coincide with traditional anatomical boundary, but some boundaries are new. And there are also a few anatomical boundaries that cannot be identified by genetic the transcriptome boundary. So this is an ongoing project, and we really hope to work with the experts in the field in the field are capturing as the world's expert on analyzing big brain segmentation oscillation studies. We hope to be able to collaborate to facilitate integration of information from mesoscopic to a more macroscopic level. Now, subcortical structure is more interesting because the gene expression are much more specific than the cortex. You can see select regions with spatial expression pattern. Subdomains of different regions are revealed. But some of these are coincide with what we expect from previous studies on clustering analysis on the whole tissue. So this is ongoing for various regions are being looked at subcortically as well as in the cortex. We can do cross-species analysis because mouse studies, the spatial transcriptome are now beginning to be available. Cell census study for human brain are also ongoing. There are a lot of data collected. So we can compare human transcriptome defined clusters, defined cell types between human and monkey and mouse. And you can see clearly there are a few primate specific. Right? If you do correlation of the cell types, you find good correlations between human and monkey. Maybe this is monkey, that's human. I'm not, there are clearly cell types that's not seen on the correlation between, there's no correlation between the monkey with a mouse, for example. So there are clearly primate-specific cell types that are revealed by this transcriptome analysis. There are also the distribution of various genes. They are very different from the cell type exists in both the monkey and the mouse, but their location in the brain are different. For example, I think this most striking thing is this FOXP2 expression in layer 4 neurons in monkeys, but in layer 5 and 6 in mice. Just an example. There are many other examples of specific monkey specific layer specific cell type distribution. And all this can be further studied. So now comes to the question, how we define cell types. Now I want to mention the difficulty of defining cell type from the subtypes, from the substate of neuron. It's well accepted in the field that major neuronal types are stable and are defined early in neurogenesis by expression of specific set of transcription factors, which define transmitter, receptors, transporters for a particular neuronal type of excitatory neuron, inhibitory neuron, modulatory neuron, all have a different set of transcription factors defined early. This is so-called ground state defined by Trace and Nat Hams. This ground state defines the major neuronal types in the brain. But what about the subtype? How do they develop? Now, I think it's reasonable to expect that these cell types are determined after the neuron is born and during the neuronal migration based on neuron glial interaction, based on the formation of genetic factor in the environment, activity included, that will set a different expression pattern of the neuron. That's what we are defining by transcriptome as a set cell cluster. Now, these are marked by if the subtypes we define there are subtypes, there should be a set of genes that are stably expressed throughout the life of the cell. And there should be relatively stable morphology of the cell type. and relative stable physiological property, although physiological property is very susceptible to epigenetic influence, especially activity, right? We can change the physiology systemically even for a prolonged period. So physiological property may not be as stable, but input-output connectivity, which give rise to the physiological firing property, these are rather stable in the mature brain. So I think the goal to define this neuronal subtype would be looking for a specific set of genes that are stably expressed together with their stable input-output connectivity. In fact, I think the input-output connectivity during development play a big role in defining the set of genes expressed through the anterior gray, retrograde exchange of factors between neurons, pre and postsynaptic neurons. So the definition of stable expression pattern is closely related to the input-output activity, which are stable after maturation. So I think that's if we can define this characteristic, this feature, we can then define the subtype of the neuron. Simply on transcriptome, that's not enough. We need to have more stable property like morphology and the connectivity. So based on this, we can then define what's the neuronal substate. That's the state, reversible state of gene expression profile. Now that, and for instance, when you do the analysis of transcriptome, there may be one pattern in a different condition, so those may be reversible. So in order to really define the stable pattern, we need not only spatial transcriptome, we need a spatial temporal transcriptome with time, longitudinal time domain information to be obtained. And that will give us the functional property that are stable for this particular neuron. And this even with a stable input-output connections, the transient activity, transient state could be different. And the neuron is undergoing short-term memory or even long-term memory, the expression pattern may be changed, but the activity may not change. In fact, there's a very good evidence that in a mature brain, the memories, the long-term plasticity involves the changes in the synaptic function and local synaptic modulation rather than long-range wiring of the circuits. So input-output long-range wiring should be a stable property. So this is, I have expanded this idea how to define this, how to find the definition in a recent article I wrote in Cell. You might be interested to look it up. It's in a comment. Then with that, the importance of connectome, I have mentioned, we really need to know for a particular single neuron, how are they connected in the brain? So there are technologies now available for single neuron tracing. So here is one using the so-called section, thin section, three-dimensional tomography or reconstruction type tomography. You can get a brain injected with the virus, then you fix the brain and section into 10 micron sections, and then you do block face imaging of all the sections. Image section, cut it out, and the next section, scan and image, and with a different probe, fluorescent probe to look at the neuronal staining, and then you reconstruct them by casing. Now, this casing takes a lot of work. So far, it's a semi-automatic huge crowd based on human and machine interaction to trace out the individual neurons. Here are the examples of tracing that you can you have to trace along all the branches, all the patterns, including the uprising at the target regions. And the automatic the tools is getting better and better, so it's getting really fast. In the past, it takes a few hours to trace a single neuron. now it can be reduced by tenfold by the most recent methods. So the first really large-scale tracing was done recently in Shanghai for mouse. They traced the 6,000 prefrontal cortex neuron labeled by GFP for different brain regions. And these 6,000 neurons, we can now cluster them based on their projectome, how their branching pattern, and how their target, or what are their targets in the brain, long-range targets within the cortex or the outside the cortex, based on target information, branching pattern, cell morphology, including dendrites. You can cluster into at least 64 different subclusters based on connectome. And this is grouped into very nicely into three major neuronal classes we know, the IT neuron, CT neuron, PT neurons. Okay? They have a very clear target region specificity for all these subtypes. So it's define the major tracts. Have more information, we can know each type, which are their targets, how their branches innovate different targets. Now, there are many interesting things. The paper has just published a few months ago in Nature Neuroscience. It has one of the most interesting things to me is that since you have the cell body location, so you will have a very clear mapping of the soma location relative to their projection targets. So soma distribution for IT neuron, for example, IT neuron have many subtypes. These subtypes are not randomly distributed within the cortex. Rather, they are clustered into different subdomains in the prefrontal cortex. Right? This happens not only for IT neuron, for PT neurons. This is the example of IT neurons and also PT neurons the same way. They also have a clustering based on their cell body. It's nicely organized. And you, based on you can find another interesting rules. That is the target region, the axon on same type of neuron. The axon arbor at the target region are sometimes segregated in different domains, and the topographic relationship in the target region matches their soma location within the cortex, right? So the soma location are actually preserved throughout the axon tract. This preservation of the single neuron neuron relationships between adjacent neurons, and that defines the close relationship of their target. For example, the branches, the axon that branches early tend to innovate targets which are closer. And then they have the target arrangement are actually related to how when they branch their axons. And then the look at the sections, individual sections through the axon tract, you will able to see that they are actually not randomly distribution. There's a topographic preservation of the tract, of the relationships in the brain. Now, you can certainly find out the rules of whether a single neuron projects from in previous study where we spoke labeling, when we look at the target region, we don't know whether this group of neurons, every one of them, are innovating multiple targets, or they are different subset of the neuron that are destined to a subset of the target, or even to a single target. And this could be now well defined by looking at single neuron projections. There are many details that I will not go into further. So I want to say that how that was in the mouse, right? So we want to see how we can extend it to big brain. Now this work is started in the last 2 years by two different technologies. First is using clearance technology to clear the large brain. We have to slice the brain into blocks and then clear the brain so that they can be used for light sheet imaging. Light sheet imaging has to be improved so that they can do a much faster imaging throughout the brain at a high resolution. Right? So this is the so-called VISOR, new modification microscopy. And then one can then trace the individual, also trace individual neurons in the brain. Now, in this example, this group of Bee's laboratory injected the dye in the medial dorsal thalamus and look at their projection into the cortical prefrontal cortex and hope to trace. And this is not sparse labeling yet, but even with this approach, they can trace some neurons within this block at the resolution of 1 micron times 1 micron times 2.5 resolution. And 3 different labeling can be accomplished by this technique. That for the cell location mapping for the morphology and cell distribution, and then specific mapping can be done with fluorescent. So now at this moment, we are only expressing this in the excitatory neuron, projection neuron. Now this is still very long time for imaging. You have to image an individual block, then stitches this block together, and also correct the slight different morphological change during the clearance process. Matching is still a challenge. And for the raw data sets is very big, about 700 TB. It takes hundreds hours per channel for recording to the entire brain. Now, this is when you get this image, you can trace. Now, this takes some effort to trace individual with this brain. This initial work was done on rather dense labeling, so it's hard to trace. Now we have more sparse labeling, so that's make this task easier, and automatic tracing become possible. So even with this, they were able to do quite a few neuron tracing in this cortex. Now this is one technology. The other technology is this technology is called a VISOR technology. The nanotechnology has been used for mice for a number of years. This involves just fix the entire tissue, inject the dye, and then fix the tissue. Inject a cell type specific, hopefully, neuron. And in most cases, the cell type expression, cell type or specific cell type are labeled. Then you fix the cell and embed the entire brain, and then section brain in a block phase through tonic map of the other cell and the morphology in different regions. It will help very much the identification of the cell location. The spatial resolution is higher because this is a higher microscopy technology, 0.3 by 1. The data is up to a PT level. So the data storage and compression of data is a very important challenge now. This is the labeling of single neuron. You can do a single neuron tracing and do reconstruction. So the other technology that's being used that's being developed in Shanghai in our institute. That's a transgenic monkey. We call it true monkeys because we want to follow the example of mice connectome. We want to label easily cell types by technology of clean monkey technology, specific types that the entire monkey brain would express specific tree in in the bronchi, and then you do local injection of particular region that you want to study, and the monkey can be reused for different studies for different brain regions. So we have generated a transgenic approach, promoter-driven PV using knocking methods, and also for manipulation technology used for studying function in the monkey brain. We have a genetic approach to transgenic monkeys. Now, a number of monkeys has been born. It's not bad. We have several have now reaching 2.5 years old. We can now doing real studies on these monkeys. There are another technology that will, before we can do real tracing, we really need to cell type markers for the big brain, for the monkey. So we have, based on the spatial transcriptome analysis, we will have specific gene expressing particular marker gene express a particular cell type or cell clusters. Then then the next step is to identify the promoters, enhancers. And then there is an enhancer screen ongoing to identify the specific marker for different cell types, region or layer specific cell types in the monkey brain. This screening methods is very successful for mice now. We can have a specific enhancer that express GFP in specific layers. Now we are applying the same technology using the same screening for monkey gene enhancers and inject them into different regions of the monkey brain. And then hopefully we can express GFP probe in specific brain region. Then this would allow us to the tracking of the tracing, single neuron tracing, as I described previously. So the hope for this entire study, I think I have my top screen blocked. Is that true? Anyway, the hope is to do the analysis of cell type determination by combining transcriptome and connectome. Single cell profiling of neuron in all brain regions, and then identify specific high expression cell markers, and then the regulatory elements, and then using single cell connecting analysis. Now, once you have a marker, that marker may not mark a connecting specific group, so you have to do recheck and doing record relabeling from the target region to relabel this neuron, and then to cell transcriptome on this labeled neuron in order to subdivide them into real target specific subtype. Right? So this have to reiterately to check between transcriptome and connectome in order to really define the single cells. And the eventual identification of stable markers for neurons that with a specific input-output pattern. That's if we get that stage, then we are really in business for functional study, right? We can manipulate a particular group of neurons, and there are really specific actions, functional activation of their targets, and then to really dissect the circuit properties. So now, eventually, certainly, we want to play a modulated circuit that modulate the function, right? So this is a difficulty for causal analysis of an interconnected network in the brain, like this, right? So this is we have anatomical studies have clearly shown that the core circuits are very clearly, easily identified in the early stages of visual pathway, and also in the final stages of the output. In the the output region and input region, you have circuits that are easily identified, whether they are core circuit. But if you go into the deep in the brain, above the association cortex, everything is connected to everything else. So now they are not equally connected, certainly. So that's why we need to figure out in all these brain regions, what are the specific cell types, how asymmetric are these connectivity between the brain region. And then to do this causal analysis, we have to pay attention to the fact that perhaps there's no clear causal or cause circuit within this complex network. Every part of the complete network is modulatory to each other, and this recognition, I think, is we have to face. This is the problem we have to face, and I think we have to all these studies I know in mice, currently optic genetic studies in mice, looking for structural circuit basis of function. The basic assumption is that by manipulating the circuit, you can up or down regulate a circuit activity. You change the up or down regulate the function. Then you say we have identified the circuit responsible, and that assumption is questionable if we think that if you modulate the manipulate the modulatory circuit, you can also up or down regulate the activity. So there are other criteria, much more refined criteria in terms of how much you manipulate, how physiological is your manipulation, and what is exactly the timing of your modulation and the activity of timing between different brain regions. All these are really important information that are lacking for most of the functional studies in the mouse. And information on our connectome to therapeutic purpose, to perform a therapeutic modulation. Because we don't really know the circuit. How do we do modulation? Most of the neuromodulation tools, whether it's deep brain stimulation or the non-invasive stimulation, they all are quite non-specific relative to the neural circuit. Now, how can we improve or the therapeutic use of these tools without even knowing the circuit, underlying circuit? Have to wait for another 20, 30 years before we work out the circuitry of the big brain before we can neuromodulation. So my short answer to these questions is no. I think we can do neuromodulation better now by thinking about several issues. One of the issues is how to figure out how does this neuromodulation tools work. How we can use the monitoring versus feedback or tools. This is a main message. I'm not going to be able to go into detail. I should say quickly that I'm particularly interested in the fact that the neuromodulation is based on plasticity of the brain. So to understand the plasticity of the modulation-induced plasticity, we have to be really clear that the plasticity depends on the timing of pre and postsynaptic timing. Now, the same closely related excitation of pre and postsynaptic neuron, all 10 milliseconds apart. If the order of excitation is reversed, you will get LTP versus LTD. So if you have a neuromodulation tools that loosely excite, be able to facilitate potentiation versus depression, you have to find a way to trigger the circuit, its own activity, to use the neuromodulation as a tool, which is unspecific, to inhibit the spiking of the naturally occurring spiking, which has intrinsic correct timing. So that's actually is my main message. Is the use of to be aware that the spike timing dependent plasticity has to be considered when we apply neuromodulation tool. Now I should just jump to the end because I think I need some time for discussion. I'll show one example, a simple experiment demonstrate that we have a motor learning by a mice on running the motor. We don't know we cannot manipulate specifically the motor learning circuits in the motor cortex because that we don't have a specific tool. But we have the TDCS, which is non-specific. So now we see how TDCS can promote motor learning, right? So the experiment shows that if you apply TDCS right at the time where the mouse is running the motor neuron, right? We call it online total stimulation. You can promote the learning greatly compared to compared to the case where you apply the same neuromodulation where the mouse is not running the motor neuron. So the promotion of motor learning can be achieved by non-specific polarization of the neurons and inhibit actually by cathodic stimulation, which inhibits the neuron. Right? So why? How can this work? Right? The idea is that, and also this effect is class specific. When you promote the motor learning, that learn the motor rod, that learning does not translate to a beam walking. Right? So this apply online TDCS when they are doing motor rod, then motor rod learning is promoted, but beam walking ability are not promoted. Right? So the specificity can also be achieved by non-specific TDCS, the total TDCS stimulation. So the basis is this. Now, if you look at this activity by two photon images, when you do a cathodic TDCS, the neuron, when the activity is fired, neurons fire on their own, spontaneous firing, the circuit is firing. But when they are walking, they are firing different patterns from there when they are at rest. So when you apply anodal stimulation, the firing pattern increases, both pre and post-synaptic, presumably. These are the neuron that are fired voluntarily by the mice. You do total, the activity is inhibited. That's expected. The TDCS change the membrane potential, so promote the firing of endogenous currents, endogenous spiking. So the idea is that now the class-specific neurons are selectively enhanced by TDCS, another TDCS, compared to class unrelated neurons. Right? So this is how the motor circuits are promoted. So when they are activated by themselves, the specific circuits, we just help them by neuromodulation at the right time. Right? So the spike timings are all preserved, but they just fire in a higher frequency, both pre and post synaptic. Right? So that's how it works. Now this can be used to correct the stroke-induced motor disability by these online stimulation methods. So this reached my conclusion for this part. Class-specific training is what we all know for a long time, that a repetitive activation of pre and post-synaptic neuron specific circuits initiated by the voluntary or forced action that can restore original connectivity or shape the formation of new connections. This has been used in rehabilitation of the. So the idea of the physical modulation, non-specific TMS, even for deep brain stimulation, is that the targets are usually non-specific, general excitation or inhibition. But if you apply that, activate that by monetary and physiological or even psychological activation of the circuit, and at the same time promote this circuit activity, either enhancing or inhibiting by neuromodulation. Then you achieve specific action of neuromodulation. So now for this, I concluded with the following thinking about the neuromodulation. For neuromodulation really to work well, we need appropriate animal models, particularly non-human primate models. for preclinical studies, all this modulation approach. Now, we are aware of this. I don't need to expand. Now, without knowing specific dysfunctional circuits, the cause of neuromodulation, for the effects we observe, then neuromodulation can modify the effects. For example, the circuit dysfunction produces dysfunctional activity. If you modulate the effects of a complex circuit, the effects may in turn modulate the cause. There's a causal effect relationship in the complex, in the higher brain area interlinked. So we don't have to find a pathological origin of circuits. As long as we have a circuit function that's dysfunctional, we could be able to promote the recovery by just modifying the effects. I mean, this is exemplified by the recent finding on the AD mice, right? This 40 hertz frequency oscillation found in AD mice are reduced. This is work by Li Chai and Ed Boyden. Now, if you enhance the 40 hertz oscillation by sound, auditory, or visual stimulation, you not only increase the 40 hertz oscillation, but you also cure or greatly reduce the cellular pathology in the AD brain, reduce the plaques, reduce the microglial activation, even neurodegeneration are reduced. So that's an example of manipulation of the effects end up changing the cause. This is because all these phenomena are also causally related, reciprocally related, right? So I think we need a systematic study of neuromodulation parameters, the strength, the timing, the duration, and the site of activation. I have illustrated today the importance of timing, the one aspect. That is the neuromodulation concurrent with the patient initiation of the target behavior. This is an important aspect, neuromodulation. And also individualized modulation. If we can monitor the using modeling approach, look at the brain dynamics using our monitoring, and then use that to feedback to do our modulation. And this for the especially non-invasive modulation. We don't really know, given a different skull anatomy and whether our modulation tools are actually working. Right, so we need to model the electric field inside the brain, and using an online monitoring the effects of neuromodulation, and then use that to tune our neuromodulation in order to achieve better action. And so online monitoring of brain state in terms help the setting the parameters. So I think I should stop here. I apologize for not long. Okay, so thank you very much.